Thank you. I did forget my disclosure slide, so for purposes, I have nothing to disclose. Now, gene therapy is something that we've all heard of. You know, you've read it in the newspaper somewhere, hear it on the late night news. In fact, in 1999, and how do I advance? Oh, there we go. In 1999, Time magazine coined gene therapy as really the future of medicine. Now, on the left-hand side of the screen here, I've given the definition of gene therapy from the Webster's Dictionary. However, simply put, gene therapy is really the use of DNA as a drug to treat disease by, delivering therapeutic DNA into patient cells. Now, to really appreciate how gene therapy has evolved, let's take a look at the timeline and the progression of gene therapy. Now, we can't really talk about gene therapy without really going all the way back to 1944, when Avery and his colleagues, McCarthy and McLeod, actually identified DNA as the genetic carrier information. And it was 1953 when Watson and Critt, really based on the work of Ross and Franklin, they cited in their published Nature on the double helical structure of DNA. And really, over the next few years, from 1960s to about 1970s, was when the term gene therapy started getting thrown around. However, it really wasn't until 1972 when Friedman and Roblin actually published their paper in Science, hence they got the credit of being the first to really cite the use of good DNA to replace defective DNA in those with genetic disease. And it wasn't really until almost 2 decades later when the first approved gene therapy took place. This was, of course, by Dr. French Anderson, pictured here with his subject, and a young lady who was 4 years old at the time by the name of Ashanti De Silva. Now, Ashanti suffered from a disease called severe combined immunodeficiency, really better known as the bubble boy disease, where she's so immunodeficient that any slight infection would be detrimental to her. This trial was really fairly successful because she was able to lead a fairly normal life, and she had to go back for maintenances, but considering she actually did relatively well. However, in 1999, just 9 years later, a young man by the name of Jesse Gelsinger, he passed away from participating in a clinical gene therapy trial, and his death caused significant setback to gene therapy research in the United States. At the time, there were a lot of people threw a lot of stones really at gene therapy, citing that, hey, it was overhyped, it's really not what it's set out to be. However, just a few years later in 2003, and this actually occurred in China, we have the first gene therapy approved for clinical use. And they actually called it Jingdesen, like medicine except with a G. And what this Jingdesen is a recombinant human adenovirus vector that actually expresses the P53 gene. And it's intended for people with tumors that's secondary to mutated P53 genes. And then a few years after that, really between 2006 and 2011, we have this huge explosion, over 1,700 clinical gene therapy trials with citing initial successes in everything from retinal disease to color blindness to Parkinson's to multiple myeloma. And of course, more recently in 2012, we have the first gene therapy that's approved for clinical use in the Western world. This product is by the name of Glybera, and it's indicated for those who have something called lipoprotein lipase deficiency. It's a rare genetic disease that causes excess of fat in the bloodstream. So really, as far as the evolution of gene therapy is concerned, it's gone through quite a bit. But how does this really translate to wound healing? So let's talk about the why and the what and the how of wound therapy and gene therapy and wound healing. Well, when we're looking at this, what we're really looking for is genes that expresses growth factors, cytokines, antimicrobial peptides that's really going to help the wound heal. Why do we do this? It's pretty obvious, because gene therapy allows the targeted local delivery of greater concentration of these growth factors and cytokines that's going to facilitate healing. Well, how does this take place? The how part gets a little complicated, so in the next few minutes, I'll try to explain this a little bit. So basically said, when you're looking at gene therapy, there's really two ways that we can introduce a gene into a cell. One is in vivo, the other is ex vivo. What in vivo is really the direct introduction of genetic material or DNA into the cell. Ex vivo is when you isolate and cultivate the cells that you have an interest in, and then transfect the cells in vitro and then implant them back into the body, sort of like in vitro fertilization. Now, whether it's in vivo or ex vivo, what's really important is the vectors that we choose to really introduce the genetic material into the cell. Now, I've listed here some of the more common ones that's used. When you were looking at it, the most classic vectors that's really used is the virus. As we all know, the virus actually infects its target cell by injecting the DNA into the cell. So in gene therapy, what we can do is, how do I reverse on this? Oops. Sorry. I'm having technical problems here. Oh, there we go. Okay, so what happens with gene therapy is basically we take out the DNA from the virus, replace the portion of the gene with the ones that we want to replicate, inject it back into the virus, and have the virus infect the target cell, hoping that the virus is going to start having the target cell express the genes that we want it to. Now, there are many, many viruses out there, everything from retrovirus to adenovirus, lentivirus, cytomegalovirus. All these virus vectors actually differ in the types of cell they infect, the amount of infection, what they do, whether it's temporary, whether it's permanent. And adenovirus, for example, is used very commonly in wound care in a lot of studies because it's shown to have a 95% efficacy rate. And however, despite all these evolutions and better understandings of the viral vector, it's still not considered 100% safe. It's almost like entrusting your best friend in the world with, $10 million and asking that your friend to deposit into your bank account and hoping, keeping your fingers crossed and hoping that they'll do what you tell them to instead of disappearing with this large amount of cash. Now, other ways that we can introduce the gene therapy into the wound is really utilizing mechanical force with high pressure. There's things called gene guns that you can introduce it. So you're actually directly introducing it into the cell using this force. Others have actually tried injection of the naked DNA plasmids intramuscularly. Now, by injecting this, some of the basic studies have found it to be of a little lower efficacy. And one way of thinking about introducing these DNA plasmids is these things are fragile. So if you're looking at the cell membrane, really it needs protection in order to get into the cell safely and harmed. So one of the newer ways that folks are thinking is using something called liposomal vectors. Now, how this works is very similar to when we wash our hands with soap and water. The plasmid DNA of what we want is actually enclosed in the center, and it's surrounded by these cationic lipids. So the hydrophobic side goes on the inside, and the hydrophilic side stays on the outside, almost like how we form micelles when we're washing our hands with soap. And by using this, it allows really the safe delivery of these plasmids into the cell to hence express the genes that we want it to. And when we're looking at these, a lot of the studies actually can utilize one or really a combination of any of these type of delivery systems. However, when we're looking at it in wound care, we must take a step back and really understand that wound care is a complicated multi-step process. You get all these mediators, I've got a diagram here. They're actually being activated, inhibited at multiple different times. So it really poses a challenge when we're looking at gene therapy. Now, to better understand this, what we did was we actually recruited patients who's got these chronic wounds. So they had to have wounds that were at least 4 weeks in length. So what we did with these wounds was we sharply debrided them. Prior to that, we actually washed them carefully and then irrigated them copiously with normal sterile saline. And after the debridement, we chopped these samples into little pieces and preserved them in Alprotect to protect the mRNA, DNA, as well as protein. We then extracted the mRNA using this Quiagen fibrous tissue extraction gate, followed by DNA synthesis and PCR array. And the PCR array that we looked at included stem cellness as well as angiogenesis, so this huge panel, okay, of 84 genes that we were looking at. And we compared and then we looked at the patients. So these patients come in and we would treat them as we normally would. If they qualified for advanced type of modalities, that's what they would get. And we treated the patients and we tracked these patients for about 8 weeks. So these chronic wound patients, if they healed within the 8 weeks, they were considered healers. And those who did not were considered the non-healers. So comparing the people who healed with the help of advanced therapy to compare them to healthy controls who donated their tissue, we're able to get a logarithmic graph like this. Now, if you look at the graph, the black line that you see right down the middle, what it indicates is that particular gene has equal amounts in the wound patient versus a healthy control. However, we are all individuals and we expect individual differences. So what's considered anything that's a 4+ fold or a 4- fold? So what you see within the yellow bars is considered of normal deviation or normal variation. So all these black dots represent genes that's really within normal limits. In other words, what we're really interested in is the genes that's upregulated versus the genes that's downregulated. The upregulated genes are represented by the red dots, and the downregulated genes are represented by the green dots. So what's interesting about these wounds, if you compare them to healthy controls, is look about the red dots. You only see 2. And remember, we had this panel that's 84 genes. These 2 genes actually did not pertain all that much to the wound healing. So really, what's interesting about these wounds is they're very similar to all the animal models that we see that's out there. These wounds are all deficient in what's required to heal. Everything from epidermal growth factors, fibroblast growth factors, tumor necrosis factors, and it actually makes a lot of sense because with the use of advanced treatments, we were able to bring these wounds onto healing. Well, now what about these diabetic wounds that actually did not heal? Let's compare their genes and what happens. So unlike the ones that ended up to healing, now we are looking at a lot more red dots up in the overregulation part. So when we analyzed these genes, what we were finding is these wounds that failed to heal had such high inflammation. It was unbelievable. You had, for all these interleukins, they were up hundreds of folds, the matrix metalloproteases. And if you looked at the genes that were downregulated, again, the epidermal growth factors, fibroblast growth factors, these growth factors, similar to the ones that healed, were downregulated. However, it seems as if these inflammatory cytokines were preventing these wounds from healing. The other thing that's interest we noted is really the deficiency of this gene called collagen type 4 alpha 3. Now, this gene actually produces type 4 collagen, is really responsible for the building of the basement membrane, and that gene seemed to be deficient in the wounds that actually did not heal. And when we actually compared the wounds of the wounds that healed versus the ones that didn't, again, the results were somewhat astonishing. The wounds that did not heal time and time again showed these high inflammatory cytokines. The interleukins were up hundreds of folds. Interleukin 6 were up thousands of folds. And it made for a very inflammatory environment that is very, very difficult to heal. And again, if you looked at the genes that were deficient, all the growth factors, the collagen 4A3, they were very they were all downregulated as well. Now, I know this is a very complex chart, but what we do is, if you plot down the 84 genes that we compared, anything that's a blank white, it really shows it's within normal. If it's downregulated, you're going to see green, and if it's upregulated, it's going to be red. So if we actually plotted a graph of these patients by how much variability or how much imbalances these patients had in their gene, the more colorful their chart was, the worse healing that they had. So it's something that's very interesting if you look into the wound itself. So what does this really mean for gene therapy? Well, we need to, in addition to looking at it, we need to identify factors that's detrimental for the introduction of genes to help these wounds heal. And then you want to make it target cell selective. The other thing that we want to do is really remember, wound healing really isn't one phase. It's not static. It's really a combination of these overlapping phases. So we need to have transcriptionally competent genes that are turned on and turned off at desired times. So one of the things that researchers are looking at is something called a genetic switch. It allows the turning on or turning off of genes really with activate stimulators such as tetracycline, and eventually develop sequential therapeutic methods. So we're looking at the transfection of multiple growth factors because that's what they're needed at strategic time points in wound healing to get these wounds healed. Now, I'm actually going to talk a little bit. I know Dr. Guntner talked about stem cells. So I'm going to talk about stem cells next and how does it play. So this is a clip from Family Guy where Peter sustained after a stroke. So he walks into a stem cell clinic.
How long was I in there?
About 5 minutes.
Why are we not funding this?
So stem cells, I mean, eventually it does seem very promising, but, are we going to heal these wounds in 5 minutes? That's what we hope to do one day. Stem cells, as Dr. Guntner mentioned, it's stable after many passages. You can have many sources of stem cells, including bone marrow, the skin, the follicles, and the stem cells are pluripotent. They can turn, for the most part, into really what you want it to. You put it in adipose induction media, and it turns into adipose tissue. You put it in osteogenic induction media, and it turns into bone. And this is actually, I'm going to next talk about the work of our PhD student, Laura Shin, who actually obtained her PhD, and this work was recently published. Now, what she did was she actually looked at stem cells and wound healing. And if you looked at these different graphs, the very top one is a wild-type mice, so non-diabetic mice that got vehicle. So that's typical healing. You can see you get day one. Now, the little splints that you see, what happens is rats or mice, they heal by wound contraction, and the splints actually prevent them from the wounds from contracting. You can see by day 12, it gets smaller. Day 14, it's really small, and by day 18, it healed. And by adding stem cells to the mice, you can see that it actually facilitated healing significantly. What about the diabetic mice? Well, the diabetic mice in row 3 that got the vehicle, you can see that the healing is significantly slower because the wound is still a fair size at 2 weeks after wounding. And then the diabetic mice that got the stem cells, you can see that at about 2 weeks, it seemed to heal. If we really looked at a plot, okay, of how these wounds heal, the yellow represents the normal mice that got the stem cell, so as you would assume, it healed the fastest. And the red line represents the diabetic mice that just got nothing but vehicle, so naturally it healed the slowest. What's interesting is really the intertwining blue and green line. So the green line represents just the regular mice that got vehicle, so normal wound healing, and the blue line represents the diabetic mice that got the stem cells. So it seems as if by adding stem cells to these wounds, diabetic wounds, we're able to help them heal a little bit more like normal wounds. But what's interesting about this is Laura actually followed this a little further. So what she did was she actually got more mice and she created 2 wounds on the back of the mice. And for the control, they both got vehicles, both wounds got vehicles. For the experimental arm, one wound got the vehicle, the other wound got the stem cells. The stem cells here is actually tagged with green fluorescent protein, so you can see it. And she followed it out for 15 days. So what's interesting, if we followed a little bit, so column 1 here is the vehicle. The vehicle, no stem cells, so you don't expect it to light up, and day 5, you don't expect it to light up. But what's interesting is the wound that got the stem cells. So day 1, it lights up very nice and pretty, but day 5, the stem cells disappeared. So wait a minute, we sit here and we talk about all these stem cells and how it affects healing and how it's supposed to heal, and we saw how it healed. Where did it go, and why is it not doing stuff that we're like? What happened here? Well, looking at the flow cytometry data actually will give us a little insight into this, and I want you to eventually focus on quadrant 4. So looking at the top graph here, this is the diabetic mice that got the vehicle. Quadrant 1 represents the human engrafted stem cells. cells. Remember, they got vehicle, they got nothing, so it should be blank. However, quadrant 4 represents the endogenous stem cell population from around the area. So in other words, by just cutting or wounding or debridement of the wound, it actually enables the honing or recruitment of the endogenous stem cells to the area. However, let's take a look at the two graphs at the bottom. The left one being the diabetic wound that got the vehicle, the other one that got the stem cell. And again, after 5 days, we saw that the stem cells, the humans engrafted stem cells left in quadrant 1. But what's interesting is in quadrant 4, notice how it lit up like a Christmas tree. So it seems as if by engrafting the stem cells, it actually helps as a signaling molecule to really cause the significant amount of stem cells from the endogenous area to come to help this wound. And it's really the endogenous stem cells that's healing the wound, not what we implanted in the first place. And in fact, it's such a powerful signal that not only did it call for its own site, the vehicle site on right next to it, adjacent to it, actually had this huge amplification of the endogenous stem cells as well. So this is interesting, but how does it really play into what we know? So by going back to our patient data for the non-healers, remember they had all these inflammatory cytokines that's around. They had a decrease in the growth factors. Well, when looking at the stem cell factors, they also have a decrease in stem cell factors. So what could that relate to? It means that we really need something more, because the addition of stem cells, yes, they will help recruit the endogenous, but you don't have a lot to start with in the first place. So this actually kind of leads into the next concept, the future concept, which is the combination of stem cells with gene therapy. Now, these guys out at Hopkins actually did a study where they looked at this protein called hypoxia-inducible factor 1, or HIF1, and how that actually affects the migration and honing of the stem cells. And they found that as we get older, there's a deficiency of this protein, which eventually leads to deficiency of stem cells. Hence, as we get older, it takes us a lot longer to heal. So what they did was they actually, using gene therapy, added these H1F protein to the wound, but they found that to be insufficient. But they combined that with the stem cells and found that to work quite as well. And some of the other studies are really looking at this future direction, which is the combination of the two. So you're looking at isolating the stem cells, and along with that, concomitantly, the generation of recombinant plasmids. And really using that ex vivo technique, you're transfecting the stem cell so that in addition to doing what it does, you have the exogenous gene expression, and eventually combine it with the development of the sequential therapeutic method to help with healing. And I just like to end with the acknowledgement of all those who work in the stem cell lab with me. Thank you very much.